Hello, we are Group 29.
And I am Daniel.
I'm Vivi. I'm LV.
I'm Chris.
A fun fact.
Now let's dive into the real talk. At the start of this journey, we were introduced to the Design Build 1 project, a device for measuring cell growth. From the very first days, we got to work exploring 3D design, microprogramming, lab work, and data analysis. We were handed a kit containing all the essential components. We decided to name the kit Penelope, calling it Penny. Over the next 3 weeks, we brought the project to life, and now we are excited to guide you through our process and share our experience.
Now that you've seen the device, let's dive into how it works. At the core of the device is a breadboard setup, which includes a light-emitting diode and a photodiode. The LED emits light through the yeast sample, while the photodiode detects the amount of light that passes through. The code running on a microcontroller processes the signal from the photodiode and converts the analog light intensity readings into ADC values, which are then stored for analysis. The code also calibrates the readings against a baseline to ensure consistent measurements. This breadboard design, combined with our code, provides the flexibility to adjust settings and ensures accurate data collection throughout the experiment.
The project began with a clear goal to create a program that can measure yeast growth accurately and efficiently. We started with a 3D model which allows us to visualize how each component would fit together. The design included a space for the light source, the sensor, and a secure area for the cuvette holding the yeast sample. After finalizing the 3D model, we used a 3D printer to bring the design to life. The result was a compact and user-friendly device. This hands-on design process ensured that our device met all functional requirements while remaining practical and easy to use.
1,000 micrometers, no, microliters, and then we have, we're going to take 1,000 of this one.
Because what I did to move it was just like
So now it's diluted.
should I
So it's
copy
1/3 of our
or should
solution
I
and
use
1/3 of the blank. No, and 2/3 of the blank, sorry.
should be separate?
Alright, it gives us 0. Whatever, it's fine. Now, this one is the real one we remade on day 2 of the lab, Wednesday 15th, which now if we measure gives us 0.11, 0.10, whatever. Continue the video, now we get this thing, and we rub it on the gel, like so. Bro, it's okay, it's okay.
Now let's talk about what the device measures. The primary metric is optical density, the OD, which is calculated from the ADC values. OD is a direct indicator of how many cells are present in the yeast culture. The device measures light intensity changes caused by the yeast cells scattering light. As more cells grow, less light passes through the sample, leading to a higher OD value. For calibration, we used a baseline ADC value of 2,879, which was obtained using a blank YPD medium. This ensures that all OD measurements are accurate and can be directly compared to each other. Moving on to how the yeast was made, dilutions and plates, to the yeast preparation and experiments. We started by creating a yeast culture in YPD medium, ensuring the initial OD was set to 0.11. This control starting point allowed us to track growth effectively over time. We performed a serial dilution to prepare samples for plating. A 1 out of 10,000 dilution was made, and 100 microliters of the diluted suspension were spread onto agar plates. These plates were then incubated for over 48 hours to allow the yeast cells to form colonies. The colonies formed to the plates correspond to viable yeast cells, which we correlated with OD values to validate the device's accuracy in estimating cell concentrations.
To conclude, despite encountering a few challenges and mistakes along the way, which we managed to resolve, our experiment successfully tracked the yeast growth curve, clearly illustrating the lag, exponential, and stationary phase. Using the collected data, we calculated the growth rate and doubling time, which closely matched the expected values for yeast under optimal conditions. This showed that our device worked reliably and that our experimental setup was strong and effective. Ultimately, this project highlighted the union between engineering and biology, enabling precise and meaningful insights. Now, thank you for joining us on this journey through 3D design, microprogramming, yeast cultivation, data analysis, and exceptional teamwork.